Non, laisse-le. Comment ça marche? C'est où? Voilà. Et là, il faut que j'aille là. Google Groups. Merde. Voilà, tu peux leur dire qu'ils l'ont. Ok, je suis là. Comment je peux... Okay, so Lina, you can start your screen sharing and we can start when you want. And I apologize for the delay and the technical problems, even though we tried.
Sorry, Eric.
Yeah.
You need to give me
Close. Yeah, I'll do that. I'll do that. Where are you? You're there. Here you go.
Eric, est-ce que je peux ouvrir la séance?
Yes.
C'est bon? Je peux ouvrir la séance?
Oui, tu peux y aller.
D'accord, merci.
Voilà, désolé vraiment pour les problèmes techniques.
Il n'y a pas de souci.
Good morning. We all know the reason for our web meeting today, the examination of the PhD thesis of Mrs. Lina Wang in order to obtain the degree of Doctor of the University of Bordeaux. First of all, I would like to thank my colleagues for having honored me to preside the thesis jury, and I have the pleasure to present the members of the examination committee. Dr. Sylvie Dinan, Research Director of INRAE, l'Institut national de recherche en agriculture, l'alimentation et l'environnement, Versailles, is the thesis reporter. Professor Nathalie Gaveau of the University of France is also thesis reporter. Professor Zhang Dai from the Chinese Academy of Sciences is thesis co-supervisor. Professor Eric Gomès of the University of Bordeaux is the thesis supervisor. And finally, I would like to present myself, Professor Rusița Tănăsova of the University of Poitiers. Mrs. Wang, would you proceed to the presentation of your thesis work entitled Combining Leaf-to-Fruit Ratio Manipulations with Exogenous Abscisic Acid Applications to Adjust Sugar and Anthocyanin Concentrations in Grape Berry? You have 40 minutes at your disposal. Please, Mrs. Wang.
Thank you, Mr. President. Good morning. First of all, I would like to thank all the general members to accepting to everyone my work, and now I'm going to talk about it. The name is Combining Leaf-to-Fruit Ratio Manipulation with Exogenous Abscisic Application to Adjust the Sugar and Anthocyanin concentrations in grapeberry. This work is under the supervision of Ahik Jiom and Zhang Wudai. I'm going to talk about it in the following 4 parts. Grapeberry growth curve is characteristic by 3 stages. During the first stage, the berry increases its size, develops site, produce and store several metabolites such as organic acid and some amino acid. The onset of berry ripening begins from Vaheron and is marked with several metabolite changes, such as sugar accumulation and anthocyanin synthesis. Grape maturity is reached when berry size, sugar, anthocyanins reach to plateau. Moreover, optimum ripeness should contain both sugar ripeness, phenolic maturity. It is well known that the greenhouse effect results in a yearly rise of annual average temperature. Warm temperatures during the growing season resulted in increasing berry sugar content. Less total acidity and earlier harvest time. Meanwhile, high temperature could reduce the concentration of anthocyanins. The differential response of sugar and anthocyanins to the rising temperature further uncooked the berry sugar ripeness and phenolic maturity. This would result in high sugar content and low anthocyanin in berry at harvest, and finally lead to high alcohol wine with poor color and aging potential. To face this problem, several adaptation stages could taken into account. For example, change group varieties or changing viticultural areas to cool temperature. However, these ways will take a long time and are difficult to achieve. Alternatively, vineyard management practice like canopy management to change leaf-to-fruit ratio is one of the most potential repeat and efficient way to control berry sugar content. As berry sugar mainly comes from maturing leaves, it's positively correlated with leaf-to-fruit ratio. In general, a leaf-to-fruit ratio around 0.8 to 1.2 is efficient to produce maximum the berry sugar. Thus, reducing leaf-to-fruit ratio below this threshold value should decrease berry sugar. However, berry sugar also correlated with the total anthocyanins in berry. After limiting carbon supply by reducing carbon dioxide, the slope of anthocyanins versus sugars a straight line keep identical to the control group. However, after reducing leaf-to-fruit ratio, the slope of anthocyanins breaks decreased. This indicates that anthocyanins are more sensitive to the change of leaf-to-fruit ratio. Why it is so and how to reduce sugar content without too much reducing anthocyanin levels. To answer this question, we need to know about the relationship between sugar and anthocyanin. Firstly, sugar can provide the carbohydrate source for anthocyanin, offering the carbon substrate through anthocyanin precursor phenylalanine, and it can also be added on canidine or defenadine as a glycol by UFGT to form anthocyanins. Secondly, sugar can also act as single molecule to regulate several structure gene or regulated genes in anthocyanin pathway, including CHS, DFR, ANS, UFGT, MIPS. So the decrease of sugar on the low leaf-to-fruit ratio will affect the synthesis of anthocyanins. Previous studies reported that ABA-induced anthocyanin synthesis by upregulating related genes in anthocyanin synthesis pathway. We can see here some of genes overlap with those regulated by sugar. Thus, ABA may promote the synthesis of anthocyanin on the low leaf-to-fruit ratio. In fact, exogenous ABA application before Vahajron can promote anthocyanin accumulation, as shown here on Shira's berry. To know if exogenous ABA can restore the balance between sugars and anthocyanins on the low leaf-to-fruit ratio, we need to clarify the relationship between ABA, sugar, and anthocyanin. In literature, there are several possible singular pathways for ABA and sugar to induce anthocyanin synthesis. First, as we mentioned above, sugar offers carbon source for anthocyanin synthesis via phenylalanine. And sugar can also serve as a singular to related single pathway based on the phosphorylating function of hexokinase. Or by regulating the expression level and the activity of SNRK1. For ABA, it may directly regulate the genes related to anthocyanin synthesis pathway, or by enhance sugar accumulation to improve anthocyanin accumulation. Besides, ABA were proved to enhance SNRK1 activity, so ABA may also promote anthocyanin accumulation by increasing phenylalanine content in berry. At last, sugar were reported to induce ABA synthesis under some conditions. So sugar may also induce anthocyanins by promoting ABA synthesis. In this work, we addressed the following questions. Why anthocyanin were more sensitive than sugar to leaf-to-fruit ratio changes? Can we reduce the sugar without reducing anthocyanin by combining exogenous ABA with leaf-to-fruit manipulations. How sugar and ABA regulated the anthocyanin accumulation in berry. Is it ABA induced anthocyanins by sugar single pathway, or sugar induced anthocyanins by promoting ABA synthesis, or they induced anthocyanins independently? To answer this question, we designed 4 experiments using 2 systems. The first three experiments were conducted in greenhouse with fruiting cutting, using fruiting cutting with 1 grape cluster of Cabernet Sauvignon. As shown in sketch map, we're reducing leaf-to-fruit ratio by removing leaves 1 week Vacheron, and change berry ABA levels by spraying exogenous ABA at the same time. Considering the huge difference transpiration between 2 leaves and 12 leaves, we add 12 leaves with girdling treatment, which had same carbon supply with 2 leaves treatment and same transpiration route with 12 leaves treatment. The last experiments were conducted with using in vitro cultural berry with different treatment. In this experiment, we analyze comprehensively metabolites such as sugars, organic acid, amino acid, anthocyanins, ABAs. Today, I will not to talk about organic acid and amino acid. I prefer to focus on the relationship between sugars, anthocyanins, and ABAs based on the result in experiment 2, experiment 3, and experiment 4. To clarify why anthocyanins are more sensitive than sugar to leaf-to-fruit ratio, we designed the 3 treatments: 12 leaves, 2 leaves, and 12 leaves with girdling, as shown in picture. Firstly, 2-leaf treatment lead to a 57.2% reduction in sugar content, and the 12 leaves girdling lead to a 51.9% reduction compared to the 12 leaves. Well, 2-leaf treatment lead to a 87.4% reduction in anthocyanins concentration, and 12 leaves with girdling lead to a 70.8% reduction compared to 12 leaves. Thus, sugar and anthocyanins concentrations were significantly reduced by source limitation. And anthocyanins were more sensitive than sugars to leaf-to-fruit ratio. In addition, girdling resulted in nice anthocyanins reduction. We then examined the transcript level of genes related to anthocyanin synthesis. The transcript of CHS2, CHS3, CHI, F3H, DFR, ANS, UFGT, and MIB-A1, MIB-A2 were down-regulated by source limitation. The peak transcriptional level of both MIBs were delayed until 83 days after the flowering. Moreover, the higher transcript amount of CHI, ANS, MIB-A1 in 12 girdling treatment compared to 2 leaves maybe explain the high anthocyanin concentration in 12 leaves to girdling compared to 2 leaves. To analyze the possible ways affecting anthocyanin synthesis, we manipulated the concentration of phenylalanine and the ABA, which is the precursor. Phenylalanine is the precursor of anthocyanin biosynthesis. From the result, we find the 2-leaf treatment increased the concentration of phenylalanine, suggesting the source limitation induced decrease of anthocyanins were not due to the decrease in its precursor level. Meanwhile, the ABA concentrations were significantly reduced by source limitation, and the reduction in 2 leaves is even greater than 12 leaves with girdling. So the reduction in ABA level but not in phenylalanine level may lead to the decrease of anthocyanins in berry. Considering berry ABA can be input from leaves and or synthesis in berry, we analyze the transcript level of the K genes involved in ABA synthesis. As shown here, source limitation silently decreased NCED transcript and not affect NCED2. So the reduction of berry ABA content may be due to the reduced ABA from leaf source, but not the synthesis in berry. And we also find that 12 leaves with girdling increase ABA synthesis in berry compared to 2 leaves, which may be the reason why anthocyanins concentration in 12 leaves girdling berry were higher than in 2-leaf treatment. Therefore, we added exogenous ABA on berries in 2-leaf treatment when removing leaves, and increase in anthocyanins after ABA treatment. The concentration still lower than 12 leaves, means the reduction in berry ABA was one of the reasons leading to the anthocyanins decrease, but it could not completely compensate the limiting effect of carbon supply. To continue analyze the potential possible reasons, we measure the transcript level of some sugar transport and SNRK1 and find that HT1 transcript decreased at 63 days after flowering. The peak of sweet 10 transcripts were delayed by 1 week under source limitation treatment. And SNRK1 upregulated at 63 days after flowering. Thus, Source limitation affect the expression of sugar transport and SNRK1, which may further affect anthocyanin synthesis. Just to summarize this part, firstly, anthocyanin accumulations were more sensitive to leaf-to-fruit ratio than sugar. Source limitation resulted in the reduction of sugar and anthocyanin concentration through change in transcript levels. A decrease in ABL content but not in phenylalanine may be related to the reduction of anthocyanin accumulations on the lower leaf-to-fruit ratio. Therefore, sugar affects anthocyanin accumulation more likely through a single effect rather than through a pure metabolite effect. The reduced ABA caused by leaf-to-fruit ratio may be one of the reasons why anthocyanin are more sensitive to leaf-to-fruit ratio than sugars. To test if combining exogenous ABA and leaf-to-fruit ratio can reduce sugar without reducing anthocyanins, we designed 6 different leaf-to-fruit ratio combined with ABA treatment. From the picture, we can directly find leaf-to-fruit ratio manipulation need to vary with pore color, and exogenous ABA enhance the color. After testing the concentration of sugar and anthocyanins in berry at harvest in 2017 and 2018, we find that after leaf-to-fruit ratio manipulation, 2 leaves reduced 57.2% sugar and 19.6% anthocyanins. After ABA treatment, 2 leaves reduced 44% sugar and 54.2% anthocyanin. Thus, it suggests that leaf-to-fruit ratio need to the increase of ratio of sugar and anthocyanins, and ABA treatment can partially restore it. To further explore the mechanism about how leaf-to-fruit ratio and ABA adjust the concentration of sugar and anthocyanins, we firstly analyze the level of berry ABA and ABA-GE, and confirmed that exogenous ABA application in significantly increased berry ABA and ABA-GE. Moreover, ABA treatment increased the transcript of gene ABF2, NCED1, and NCED2. Then we analyzed the effect of exogenous ABA on sugars. ABA treatment enhanced the sugar accumulation under source limitation, and the increase HT1 transcript under source limitation after ABA treatment may be related to these persons. However, ABA did not affect SNK1 transcript. Exogenous ABA enhanced anthocyanin accumulation both under source limitation and under 12 leaves. transcript level of genes CH3, CHI, F3H, F3', 5'H, UFGT, MIB-A1, and MIB-A2 were increased by ABA. The transcript of ANS, UFGT, MIB-A1, and MIB-A2 peaked earlier may be related to the earlier accumulation of anthocyanins under ABA treatment. And the increased transcripts level of F3', 5'H may related to the decreased ratio of D2,3 hydrocyanated anthocyanin. We also detected the concentration of phenylalanine and found that ABA had little effect on phenylalanine. Thus, the increase of anthocyanin after ABA treatment may not due to an increase in its precursor level. To summarize this part, reducing leaf-to-fruit ratio decoupled sugar and anthocyanin accumulation in berry, and ABA particularly restored it. Exogenous ABA increased the sugar concentrations under low leaf-to-fruit ratio, which may be related to HT1 transcript amount. ABA enhance anthocyanin accumulation and affect D2,3 hydroxylated ratio by increasing the transcript level of gene biosynthetic and regulatory genes. ABA induced anthocyanin accumulation independently of the SNRK1 to BZIP11 to phenylalanine pathway. To clarify how sugar and ABA regulated anthocyanin accumulation in berry, we plan to change ABA content in vitro berry system by adding exogenous ABA or by inhibiting ABA synthesis using the ABA inhibitor NDGA, or change sugar content by adding glucose, or by affecting the phosphorylating activity of hexokinase by its inhibit NAG and DGH. Firstly, we analyze how sugar-related anthocyanin accumulation in Fig A. We can say in higher sugar content, in higher glucose concentration in cultural medium resulted in higher sugar content. And the two hexokinase inhibit did not affect the uptake, affect exogenous glucose uptake into berry. In fact, B, high glucose induced anthocyanin synthesis hexokinase inhibit DGH in decreased the concentration of anthocyanin, but NAG had no effect on it. Similar to the result in fruiting cutting berry, high concentration of glucose lead to a decrease in phenylalanine, suggesting sugar may act as a single molecular induce the synthesis of anthocyanin, and the DGH decreased the concentration of phenylalanine, but NAG did not. This suggests the DGH may affect the sugar metabolite. To test the hypothesis, according to which sugar induce anthocyanin synthesis by promoting ABA synthesis, we detected the ABA and its catabolite concentration in berry after 3 weeks of culture, and find that high glucose did not affect ABA concentration, and hexokinase inhibit did not affect ABA concentration, which suggests sugar-induced anthocyanin synthesis not due to increased ABA synthesis. Then we analyzed how ABA regulated anthocyanin accumulation. Firstly, exogenous ABA greatly induced the ABA concentration in ABA as well as the concentration of ABA-GE. And the inhibitor of ABA synthesis, NDGA, had no significant effect on berry ABA concentration. About the question whether ABA induced anthocyanin synthesis by promoting sugar accumulation, from our result, ABA can significantly increase the concentration of anthocyanins, but had no significant difference between control in sugar concentration. Thus, ABA-induced anthocyanins not dependent on sugar accumulation. In addition, the inhibit of hexokinase DGH can block the induction effect of ABA on anthocyanin. A two-way interact analysis between sugar and ABA treatment shows that both high glucose, such as in Fig B, and ABA treatment, such as shown in Fig C, can induce anthocyanin synthesis, but ABA increased the anthocyanins were higher in high glucose medium, suggesting the effect of ABA and the sugar on anthocyanin concentration are synergistic. We also studied the effect of ABA on phenylalanine and find that ABA decreased the phenylalanine and the NDGA had no effect on it, suggesting ABA-induced anthocyanins not by increase the precursor of anthocyanin. To summarize this part, both high glucose and ABA-induced synthesis of anthocyanins. ABA was able to induce anthocyanin independently of sugar content. Sugar are able to induce anthocyanins independently of ABA content. DGH decreased both phenylalanine and anthocyanin concentration. ABA and glucose-induced anthocyanin synthesis are synergistic. To conclude our result, reducing leaf-to-fruit ratio increase the ratio of sugar to anthocyanins in berry. The reduced ABA caused by leaf-to-fruit ratio decrease may be one of the reasons why anthocyanins were more sensitive than sugar to leaf-to-fruit ratio change. ABA and the sugar are able to induce anthocyanin independently. ABA and the glucose-induced anthocyanin synthesis are synergistic. Thus, combining leaf-to-fruit ratio manipulation with ABA treatment could be one way to reduce berry sugar content without too much reducing anthocyanin levels. Finally, we propose this singular pathway. ABA increased the sugar content by increasing the transcript level of some hexyl transport such as HT1, and ABA also increased anthocyanin concentration by enhancing the transcript level of genes related to anthocyanin synthesis independently of sugar content. While sugar, it may induce anthocyanin synthesis pathway through SNRK1 pathway, but the effect of SNRK1 on anthocyanin still need to be confirmed in berry. For the further work, we need to measure ABA and its catabolite level in the remaining sampling, and analyze the transcript level of genes related to sugar, anthocyanin, and ABA by qPCR with in vitro culture berries. The activity of SNRK1 on the DGH with in vitro culture berries should also be detected. There are many people who help me in this work, and I would like to thank to them here. Firstly, I want to thank my two supervisors, without whom this study would never have been possible. So thank you, Ehik and Zhang Wu. I also want to thank our two collaborators on this project, Sabine and Eloise. It's been a great time working with you, and thanks to Claude, Christelle, Duyen, Rislan, Isabelle, Jean-Pierre, Misa, and Nabil, who really help me a lot in what kind of experiments. And I also want to thank the team of CSIOR Agricultural and Food Lab in Australia, especially Annette and Everett, who helped me to analyze the ABA and its catabolite in berries. During these 4 years, I also enjoyed the opportunity to meet and share lifetime with many students. I want to thank them here, and I also want to thank my friends in China and friends and my family. Thank you for understand, encourage, and always support me to finish this work. Thank you.
Thank you so much, Mrs. Wang, for this clear, very nice presentation. Now, Dr. Dinan, the first reporter, would you let us know your evaluation of the work of Mrs. Wang, and would you proceed to the examination?
Okay, so thank you very much for this very interesting presentation of your work. So as a first reporter, I will give a few comments on the manuscript and on the presentation. So I really enjoyed the manuscript, the thesis manuscript. And there were a few details that were not so clear in the manuscript, and I'm really glad that many additional insights on sugar metabolism, sugar accumulation, anthocyanin accumulation, and sugar signaling has been provided during the presentation. And the presentation is also shows many new figures. So I really appreciate both the manuscript and the presentation that really provide a very straightforward overview of the work which has been done. So this is the point. The second point is that you have shown in your presentation a lot of data. I think that these data were obtained with a lot of work, and in the manuscript there are also many additional data that has not been presented today regarding the accumulation of other components, including organic acids or amino acids, and it's also important work, and the analysis of all these data should provide very interesting insights on the metabolism that occurs during berry maturation. So it was very interesting for me to read the manuscript and to listen to this presentation, because I really learned a lot of things. So thank you very much, Lina, for
Thank you, Simila.
The manuscript and the presentation. However, I have a few questions, of course.
Yeah, of course.
So so I have two very general questions. The first general question is about the difficulty to separate the role of sugars as metabolite building blocks for most other metabolites in a plant, especially in berry, and the role of sugars as signal molecules. And in your presentation, you showed some of the experiments that you have carried out to propose a role of sugars as signal molecules. Can you just tell us whether many other studies in the literature on other models have tried to separate the role of sugars as metabolite or as signal molecules?
Yes. I know there are some studies, maybe from the in the berry cells, and also use the in vitro cultural berries. They also do the similar treatment, such as I use the hexokinase inhibit. As they use the NAG also, and find use this inhibit the anthocyanins accumulation or reduced. So in fact, I based on this result to design my experiment.
So do you think
I'm sorry. So so they propose that sugar can, as a single molecular, to induce anthocyanins based on the phosphorylating of hexokinase. So it's because this I also want to test in our experiment. Yes, thank you.
So do you think that these approaches definitely can definitely answer the question whether sugars act as signals or act as substrates, metabolites, or what do you think of these approaches in general?
In my opinion, if sugar acts as carbohydrate source, the key point is the phenylalanine concentration, the role in phenylalanine. So if we work the way to for sugar to produce phenylalanine, the anthocyanins concentration should have a change, should have reduced. I think this way can prove sugar can act as a carbon source supply to regulate the anthocyanins. And for the sugar singular passive for sugar as a singular molecular, if phenylalanine had not changed, but anthocyanins also induced, and the genes in anthocyanin pathway also be upregulated. This can prove sugar may as a singular molecule to regulate the genes in anthocyanins pathway.
Thank you.
So, thank you. I don't know if I speak clear.
So that's interesting. You emphasize the use of phenylalanine as a marker of metabolism, rather than a marker as as a marker of metabolism using sugars as a precursor. So it's so it is also integrated in your other general figure, which describes the different components of this very complex signaling pathway. So I have another question. So maybe we'll discuss later your data on phenylalanine. So I have one more question on a general introduction of your work. So you have used in this work one cultivar, Cabernet Sauvignon, which is a red cultivar, of course. So can you tell us more on the variability of red cultivars And why did you choose this cultivar rather than another one?
Yes, they have many other grape vetiver, such as Cabernet Franc and such as and others. I choose this this one is because in our region, Bordeaux, Cabernet Sauvignon is very important. It's very important ones to produce wine. They be they have many wine maker use this one, and our in our fruiting cutting system, this one also works very well, and it can the berry developer can much good with the field much good with the plant in field. So I choose this one to analyze this problem. Yeah, thank you.
So do you think it's a representative genotype for red grapevines regarding accumulation of anthocyanins or accumulation of other metabolites in the berry?
In fact, the berry sugar and anthocyanins content varies from what kind of varieties. But this one we choose is very classical ones, I think. Yeah.
Thank you.
Thank
you. Regarding your introduction, now I will have a few questions for the introduction of your manuscript. Yeah. You describe many different pathways, so it's very interesting. So regarding sugar transport in the berry and in grape in general, you have a figure, which is a figure 3, which shows that in all organs, in the different organs, you have a combination of different sugar transporters. And this combination can vary according to the developmental stage. For example, during the maturation of the berry, you have different sugar transporters that are expressed. So this is based on the description of a transcriptional regulation of these genes, of the genes encoding these different sugar transporters. So can you tell us whether there are other levels of regulation that are not indicated in your figure, and that could be very important also for controlling sugar accumulation in these organs?
I'm sorry, I want to make sure about the question. Is if they have other sugar transport, not in this figure, but also important for berry development, is that?
Okay, so that was not the question, but I think that you can indeed make points on the other transporters that are important during berry development. But the other question was that these data show the transcription levels of these genes, and it could only reflect one level of regulation, which is a transcriptional regulation. But the question is, do you know whether the sugar transporters can be regulated at other levels than transcriptional levels?
Other levels.
Phosphorylation or dimerization or.
I'm sorry, I didn't. I think I didn't. Yeah, I'm sorry, I didn't know this. I didn't read the articles about this.
Okay.
I'm sorry.
So my question. So in your work, you have looked at the accumulation of the transcripts of some of these genes.
Yeah.
And in the interpretation of the data, if there are other regulations, for example, phosphorylation, maybe the transcriptional level will not be sufficient to really conclude to the data that you have. So I think it could be interesting to look at in the literature or the information, for example, for the sucrose transporters, such as SUT transporters, they can be phosphorylated. So looking at the transcription level may not be sufficient to have an idea of their regulation, for example, in response to sugar or to ABA. So maybe this is something in the discussion of your data, of your manuscripts, that could be just looked at. I think it's also important.
Okay, thank you.
So the introduction was describing many aspects of the fruit biology. So I will not comment all the different parts. So it was a very interesting introduction. So maybe I can switch to the different experimental part of your work, unless some other, well, so I have some questions on your work.
Okay.
So so maybe we can discuss now on phenylalanine. So I really like what you say during your previous answers. So you said that phenylalanine is a good indicator of the use of sugars for metabolism and for the synthesis of anthocyanins. So do you think there is a clear correlation, or is there any element in the literature showing that there is a correlation between the level of phenylalanine and the synthesis, the biosynthesis of anthocyanins?
Yeah, there are some studies on berries, berry cell or on berries. They use exogenous phenylalanine to application on a berry cell and or berries, and find that it can increase the phenylalanine concentration. And they also add exogenous phenylalanine and ABA at the same times, and find they have some maybe exact some interactor to induce ansosylins. So so the increase in phenylalanine increase ansosylin synthesis.
Hear us?
Probably Sylvie is frozen.
Sylvie is freezing. Maybe I sent her. Oh, she left.
She may rejoin.
She may reconnect. Yes, sure.
Yes, yes.
Lina, if she reconnects, can you admit her again?
Oh, okay. I didn't.
Okay. Can you hear me?
Yeah, now it's okay.
Oh, I'm sorry, I had an interruption in the transmission. I could not hear you any longer, and I could not see you. So I'm sorry, it was a problem.
Lina, probably you can repeat your answer.
Yes, please.
To the question.
Okay, yeah. They have some report. They have some studies which reported that exogenous phenylalanine application can increase ansosylins concentration in both berry cell and the berry. And they also study on using exogenous ABA and phenylalanine, and find that they may be exact some interactor to induce ansosylins.
Okay.
So so increasing the concentration of phenylalanine made increasing ansosylins synthesis.
Thank you. But what about the level of phenylalanine in the berries when in these studies where they apply exogenous phenylalanine? Is there also an increase in phenylalanine in the berry? Does it reflect the application of exogenous phenylalanine?
In our result, we didn't find we didn't find both ABA treatment or high glucose treatment to induce phenylalanine, and it's on the other side, it's even decrease the concentration of phenylalanine. I think it's maybe the stable state for phenylalanine during berry ripening, but as our other result, the high glucose and ABA really increase ansosylin synthesis, and it's also upregulated the downstream genes of phenylalanine. I mean the genes in anthocyanins pathway. So I think they should be a bigger role for sugars or ABA to regulate the downstream of phenylalanine genes to use phenylalanine.
So, did you try to use phenylalanine, exogenous phenylalanine in your in vitro system?
Yeah, it's we don't use, but it's a really good idea. I also think about it. Maybe we can to add phenylalanines with the inhibitor of hexokinase. Like DGH, because it broke the anthocyanins, and we don't know why. To make its more clear, it's if affect the up phenylalanine or down phenylalanine. Yeah.
Yeah.
I think yeah.
Because with your in vitro system for berry development, the data that you have obtained are not so clear about the role of sugars. Well, they are clear, but they still could be a major role of sugars as precursors for the synthesis of anthocyanins. So using phenylalanine could be
yeah,
a way
it's a good idea
to distinguish the two roles. So it could be interesting.
Yeah.
In your first chapter of results, when you describe your system, you have a very nice figure, which is a figure 1 on page 53, which shows that the leaf to fruit ratio depending on the leaf numbers on the plants.
Yeah.
And this figure is interesting because it shows that there is a huge variability from one year to the other and from one treatment to the other regarding this leaf to fruit ratio. And when you describe the data that you have obtained with your plants, you always refer to the data as a consequence of change in the leaf to fruit ratio. However, in the following figures, you show your data regarding the number of leaves that remain on your plants. And I was wondering, why don't you present your data, for example, quantification of exos or anthocyanins, regarding actually this ratio, leaf to fruit ratio, instead of presenting the data as a function of the number of leaves that remain in the plants?
Yes. It for one reason is it's more easy to describe with leaves number. Another reason is in 2014 we can say the ratio of leaf to fruit ratio is a little bit lower than other years. So we and some results in 2014, yeah, it's also have some effect, different effects we think it may be that. So we use we use the number of leaves to describe our treatment.
Yes, but you could have you could obtain less differences depending on the year if you express your data as a function of leaf to fruit ratio, because what we show in the following figures is a large variability depending on the years. So maybe you could reduce this variability if you actually use the leaf to fruit ratio instead of leaf numbers, just to limit this variability between experiments. So
yeah, I understand you now. Yeah, I think it's maybe a way to show the result. But for us, there are another problem. It's because we harvest the berry based on the berry as I shown in the presentation in introduction. We don't base on the same sugar concentration. We harvest the berry different sugar content, so it's maybe another reason to affect the sugar content or other content different from year to year. So I think even we use leaf to fruit ratio to make it may be make a big change of the treat.
okay.
Yeah,
okay.
It's for that. Yeah.
But in this case, maybe so when you discuss this data, you discuss them regarding the fruit to the fruit to leaf ratio, and not regarding the leaf number. So it may be confusing because you consider different things.
It's
The
true.
same number of leaves you have depending on the of the samples, a huge variation in leaf to fruit ratio. So be careful in the description of your data because you are not looking at the same values. So it's could also allow you to have different conclusions.
Yeah.
And in any case, it's always interesting to compare different way to present data because they can highlight different results.
Yeah, it's true. Yeah.
So actually, you did not present organic acid and amino acid results.
Yeah.
But I found this data very interesting too.
Yeah.
Because they provide a lot of information on the change in the metabolism, in the primary metabolism. So I have just two questions on this part. We are not looking at the data, but looking at the significance of this data. So you have huge variations in malate, one of the organic acid, and huge variation in proline, which is a very abundant amino acid in the berry.
Yeah.
Do you think that the levels of accumulation of these two metabolites is also important for the quality of the berry at harvest, for the organoleptic properties of the berry at harvest.
For the for the organic acid and the proline in berries, it's important to the for the berry at harvest, because it the ratio of sugar to acid is maybe the most important intact for harvest berries, and the proline content is also important for wine making, for the berries for wine making.
So in my report, I suggested that other method could be used to integrate all these data, including amino acids, organic acids, sugars, ABA quantification, anthocyanins, to extract more information from your data.
Maybe it's not so clear, but we try to. I think it's a good idea for presentation the result. So we also try to do a PCA based on the data.
Okay. So do you think these approaches should be integrated in your manuscript before publication? What do you think of statistics for this statistical tool for exploiting all the data?
We can see from the figure, it can divided the different leaf to fruit ratio, but for the ABA treatment, it can't divided it. And for this, the anthocyanins and the sugar may offer a big score on it. And I think it's added some information to for our to say more high into our result. And for the first article, we just received the review back from our journal, and it's looks positive, and we just need to modify some. We just moderate reversion, and so it's good.
So at least thank you for following my recommendation and preparing the figure. Now I will switch to gene expression. So you also have a lot of data regarding gene expression. So I will focus on the expression of genes related to sugar homeostasis and sugar transport, because as you know, I'm more familiar with that. So so regarding the expression of genes that are involved in sugar transport. You have chosen, I think, 5 genes,
Yeah.
including an exhaust transporter and several 2 sweet genes. Can you tell us why you choose these particular genes and why you didn't choose others?
Yeah, in fact, I want to choose more genes, more than 5 genes related to sugars, because based on the previous study, there are really a lot of genes related into berry ripening. But for me, I choose 2 as you say, the score transport. It's reported that they are upregulated after Vahazon, and it may important for ripening, berry ripening. And for HT1, it's because it's reported that it may involved in both sugar and ABA single pathway to regulate the berry ripening. For SNRK1, I think it's very important genes who can balance the polymer and secondary metabolites, which may involve to our study, that is sugar to anthocyanins, this pathway. And it's also reported that it can sometimes it can induce anthocyanins, sometimes it can inhibit. So I'm very interested about this, how they balance the sugar, how many sugar to offer carbohydrate source to anthocyanins. So I choose this also. For the sweet 10 is reported that have a peak in Vahazon, and it's another gene family. It's represent another family of sugar transport. So I also add one of these. In fact, if I can add more, or I can continue to do this, I also want to test the genes HT2, HT6, because it's also reported regulated by ABA. Yeah.
Okay. So why don't you choose TMT1 and TMT2, which are also based on your figure on sugar transporters, very highly expressed during fruit maturation in berry?
In fact, in my opinion, I most want to choose the genes which may both related to sugar and ABA, or something links between sugars to anthocyanins.
Okay.
So yeah, so I prefer the genes like that. Yeah.
So you were looking for genes related to sugars that could be that could be regulated by either sugar or ABA. Okay,
Yeah.
I understand why you have chosen. So, what kind of information did you obtain regarding the response of these genes? Do they provide information on the regulatory pathway, or do they provide information on the physiological events that could be enhanced in response to some of your treatments?
I'm a little lost in the question. Do you mean if the...
Yeah, do you think that your data can tell you that there is more sugars coming into the berry, or more sugar accumulation in the vacuole in response to ABA or defoliation? Do you have, what do you think?
From our results, I just find the genes sweet 10 and it's I show you the we find the two SUC they didn't be affected by sugar or by leaf to fruit ratio or by sugar input into berry. For the TH1, they have a little change, and the sweeten, I think it may be the ones who can say something about sugar flux into berries, because the peak of sweet 10 transcripts were delayed by 1 week. It's just matched to the berry content. So I think this gene may say something, but for TH1, it's high expression before Vaheron. So it's maybe not because there are more sugars being transported by it into berries, I think.
Okay, so both genes are expressed in the are located. So both proteins encoded by these genes are present on the plasma membranes of the berry.
Sorry, both of the genes.
So what is the location of these proteins? Are they located in the plasma membrane, or are they located in the tonoplast of the vacuole?
I don't understand the word
So the question is whether these two transporters, sweet-10 and HT1, allow sugars to enter the cells, or whether they are involved in the storage of sugars in the vacuole.
Both the two transport are located on top plastic, so it's transport sugar into the cell. into the berry cell. Yeah.
Okay, thank you.
Thank you. I'm sorry to for my English.
No problem. And my questions were not clear, so sorry.
No, it's me. Yeah. Thank you for repeating.
I will not. I still have many questions, but I don't want to use too much time. So I have one question on the last part of your work, which is the part on in vitro culture of your berries. So I think it's a very interesting system because you can directly assess the effects of different molecules. So in this system, you have used glucose to provide carbon to the berry for its development.
Yeah.
So in the real life of a berry, sugars are transported as sucrose. And the main source of sugars is sucrose, which is delivered by the phloem and then which is metabolized and transformed in exose, including glucose. So why did you choose glucose instead of sucrose for this in vitro culture?
Yeah. I choose glucose is based on our previous study. Before this study, we have another study to special use different sugars, including sucrose, glucose, and fructose, and also use different concentration. There are even more than me use in here. So, and we find that the glucose, the effect of glucose is the best. So in this study, we just use glucose.
Okay.
Yeah.
So, do you think that other sugars could be used, especially? Do you know whether it is possible to use sugars that cannot be metabolized with the objective to address the question of sugars as signal molecule?
Yes, we for the I say in other studies they use the many as I'm sorry, I forgot the English name, but it can be it's one can't be used in berries to they use to analyze if they can have an effect on also anthocyanin, and it's true if we use this they can't produce anthocyanins, and yeah, we can use this.
Okay.
Yeah.
So thank you. So thank you very much, Lina.
Thank you.
What I can, I will finish.
Thank you.
And I will just thank you for this nice presentation. Thank you very much.
Thank you for your question. Yeah, it's I really like the phenylalanine, exogenous phenylalanine to use. I think it's a very good idea to continue our experiment. Yeah. Thank you.
Thank you, Sylvie. Thank you, Lina. Now, Professor Gavot, as second reporter for your thesis, please let us know your how you assess the work of Mrs. Wang, and please,
Rossi?
which are your comments and questions?
Rossi, if I may just make one short intervention. People are asking if Lina can stop the screen sharing so that they can see the jury on the YouTube. I mean, Lina, you can stop the screen sharing and then you can restart it when you need to work on a specific slide, maybe. Thank you.
Okay.
Please, Dr. Gavot.
Yes. Thank you, Rosita. I would like to thank Eric for your invitation. And I will first congratulate Lina for her work and for her presentation. I appreciate the clarified of the presentation. I appreciate the summary diagram with the relation between ABA, sugar, and anthocyanin, because it's missing of the manuscript. Globally, your manuscript was easy to read. The story is easy to understand. You present a lot of results, a lot of data, but there is a lot of missing data of statistic result. For example, for all the figure in the chapter 1, you have not statistic result on the figure. I ask myself, is it was an omission or if it was no difference? But I think that is an omission. Yes?
Yeah.
Could you add a result for the publication? Because it's not good for the moment. I will take the manuscript in order, but some questions have already been taken by Sylvie. I have the same point of view that Sylvie concerning the presentation of leaf to fruit ratio. It's my first question. It's very difficult to compare your results because Sylvie say that, for example, for 2014, leaf to fruit ratio with 2 leaves is the same that 6 leaves for 2015 and 17 and 18. I think that it's difficult to compare your results if they were expressed in number of leaves. I think that it's easy to compare with leaf to fruit ratio in gram and in meter square. In first chapter, you work in greenhouse, okay?
Yeah.
Why do you make one manipulation by years? Do you have an effect by years related to reserve? It's related to temperature? Why?
I want to make sure the question you want to ask me why the result of sugar or something content different year by year.
Yes. And why do you make just one experiment by years?
One for the first Yeah, for the first question is, I think the difference year by year, maybe it's because we planting the plant in greenhouse different times, because in greenhouse we have two times to planting the fruiting cuttings. I think this is maybe one of reason, and another reason maybe it's because we harvest the berry based on the berry size, berry seed, and they are and the sugar and sustaining to not continue increase and harvest them at the same time. So maybe different year they have different breaks, which may resulted in different value, but the trait is the same. It's similar. It's not totally same, but it's similar. Yeah.
Yes. Okay.
And for for the I think for order test, I all use for the leaf to fruit ratio experiment. I do in 2013 to 2017 is 4 years, and for the ABA treatment to add to compare leaf to fruit ratio with ABA, we do the experiment in 2017 and 2018. So it's all year by year. Yeah. Thank you.
Okay. And in this fruiting in this berry, you measure different form of anthocyanin. Why? Is it an important parameter for the quality of the vine?
Yeah, we analyze the different anthocyanins and to compare the D2 hydroxylated anthocyanin and acid or no acid anthocyanins. It's because in general the tree hydroxylate anthocyanins, the color are little deep and more stable, and it's will affect the color in wine also.
Okay.
So yeah, thank you.
And concerning ABA, could you give us some information about the synthesis? In which organ, in which cell compartment, and is it transported in the plant?
For the ABA, in berries, they are it's the source maybe complexity, because at the beginning we thought it's sensitive in berry, but they have a important review for us. They said in 2016, I think. It's studied on single berries, and they really separate the different tube. The different berry begin to increase ABA, and the begin to increase the transcript of NCED 1 and 2 also, they find the increase of ABA before the expression of NCED 1 and NCED 2. And it's just maybe the same as the tubes begin tubes. So they think it's maybe from the berry ABA may be from leaves or be from the ABA-GE in berries. So in our environment, we also detected the ABA and ABA-GE and the gene NCED1 and NCED2 to confirm if the changes in genes are because the synthesis of ABA reduced or other ways.
Okay. And do you think that ABA treatment have a consequence on leaf and on, for example, leaf photosynthesis or sugar storage in stem or in roots?
Yeah, they have a study to special to analyze if ABA synthesis in root or in leaves, and I think it's published in 2018 in Nature. It said they have several PT protein respond to the water content changes. The name is CLE. CLE25. They produce in root and then transport into leaves, and the leaves to produce the ABA and then transport back to root. So in our environment, I didn't show in our result. I also try to analyze the transcript level of CLE25 and another is CLE like, but I didn't find a big change. So I'm not sure if in berries they have similar mechanism.
Okay, and do you think that ABA treatment has of buds?
I'm sorry. I just.
I repeat. Do you think that ABA treatment have consequence of buds?
I think Gavo, Natalie Gavo, maybe. I just broke the internet.
Okay. Okay. I repeat my question. Do you think that ABA treatment have a consequence of buds?
ABA 对芽有没有影响？
Yes, I just I think they have some effect, but I didn't read many articles about this.
Okay, but it was shown that the ABA has an effect of latence in grapevine. It's a study of another laboratory in ESVV. Because you know Laurence Jean-Geny? No?
Yeah.
Yes.
Yeah.
So just this question, because do you think that your experiment is transferable in the vineyard next year, for example? And if yes, how many applications of ABA? When should the leaves be removed? How many?
You do you means if ABA can use into vineyard, and how many we can use? Is that?
Yes.
Yeah. For the Europe, in winemaking, in viticulture, I think ABA were not, maybe it's forbidden to use in vineyard. But our results are many. In my opinion, it's to find some clues about leaf to fruit ratio and the ABA to affect the berry composition. If we want to increase berry ABA, maybe we can use other ways. For example, to use different water straight, or change some microclimate. They also can induce berry ABA. And for the if not for the vineyard, if not for winemaking, as I know, ABA can be used to improve anthocyanins in fresh berry or dry berry, the table berry or dry berry. Yeah.
Okay. And my last question, with your perspective, you present 3 techniques in your perspective, but what is the biological question? You present RNA, you present activity of enzyme, but what is your biological question?
For it's biological question for my study.
Yes.
Okay. For the for the future, yeah.
Yes.
Yeah. I'm very interested in the in the why the inhibit of 2 hexokinase have different effect on anthocyanin synthesis. I think for the future, I want to maybe to continue to find the target of DGH, to say if it's involved in balance the sugar and the anthocyanins pathway. And if I think it will be interesting to change it. By changing it, we can reduce sugar and maybe increase anthocyanins, not change other things. It will maybe will be more useful, I think.
Okay, and if you must choose, what would you start?
You means the candidate genes? Yeah, I think SNRK1 maybe is one of them, and another one I think is it means MPK4 is reported in other plants, which is mapping canals will to touch with MIB27, I think, to affect the anthocyanins synthesis. Yeah.
Okay.
Thank you.
Thank you, Lina. That was my last question. Rosita?
Thank you, Professor Gavot. Thank you, Nathalie. In my turn, I will discuss with Lina.
Yeah.
First of all, thank you, Mrs. Wang. Thank you, Professor Dai, and thank you, Professor Gomès, for having invited me in the thesis jury. Thank you, Lina, for your so elaborated presentation, summarizing a huge amount of results. Very nice presentation.
Thank you.
Obviously, you did very hard and impressive work. I highly appreciate the conceptual approaches going to address so difficult and complex question as the crosstalk between ABA and sugars in the gene expression regulation during the grape berry ripening. And really, it is very interesting to choose two complementary experimental systems, the whole grapevine plant in greenhouse and the grapeberry cultured in vitro, and a very elegant model tested under 21 different conditions. Really impressive. Your manuscript is well organized and very pleasant to read in a good scientific English, and I congratulate you.
Thank you.
Reading the manuscript,
Yeah.
I have a difficulty concerning the frequent absence of fully developed and detailed legends of the figures. It appears obvious that the data presented on a figure have to be understood even in the absence of explanation in the text. For example, the figure 5 in the introduction is dealing with the biosynthetic pathways of anthocyanins in grape. The precursors, the intermediates, and the final products are very well identified. But, however, the enzymes responsible for these metabolic pathways, whose encoding genes and activity are the main topic of your work, are cited by abbreviations, and their complete names, highlighting their functions, are omitted, and moreover, they are missing in our abbreviation list. I think that you have to correct this.
Yeah.
In several other cases, the legend of a figure simply relies on the legends on a previous figure and so on to a third one. I think that we have just to copy this part and place it at the right place. and each figure. Bon. Now I have some questions. Do you think that there are really two different signaling pathways, one for the exogenous ABA and one for the endogenous ABA? If so, which are the arguments to corroborate this?
For the sugar-induced insufficiency gene pathway, I show here they have two possible ways. One is hexokinase, another is SNRK1, and I think they should have other ways. But for us, we focus on these two. For the hexokinase, it's because based on previous study, there are many studies reported that sugar-induced insufficiency based on the hexokinase phosphorylating function. If we inhibit this function, the insufficiency can't be synthesis. So at the beginning, I want to make sure about it. So I choose to inhibit of hexokinase. And for SNRK1, it's because it's reported to be the center protein to regulate the balance between terminal metabolites and secondary metabolites. So as we change the carbon supply by reducing leaf to fruit ratio, I think maybe they have some effect on it. So I want to test if these genes play important roles in between sugar and anthocyanins. Another reason is in other study, it's also reported SNRK1 involved to regulate the anthocyanins pathway. By our expression, it's overexpression of SNRK1. It can block the 3% glucose-induced anthocyanins synthesis in adipocytes. So I think maybe it's also related to our study. So I choose these two pathways to further study. Yeah.
Thank you. Do you know if there is another glucose signaling pathway independent of the exokinase?
Yeah. I read an article to say it's sugars may affect ABA since a singular pathway like VVSK1, I think, the genes to affect the downstream single pathway, but they didn't say it will have effect on anthocyanins. So I didn't choose other genes. Yeah.
Yes. It is interesting to remember that there are another glucose signaling pathway independent from the exokinase activity as a cytoplasmic sensor and as from its catalytic activity. This independent pathway, in this independent pathway, there is already reported many genes regulated by this way. And between these genes, we have the phenylalanine ammonialiase, the chalcone synthase, and the cell wall invertase. 3 genes really studied in your work.
Oh.
That I means that your choice a little bit classical, your choice of transporter genes and a little bit classical. I agree that it is very difficult for the time of the PhD thesis restricted to 3 years to enlarge the study, but it will be very, very interesting to study other transporter genes and maybe use inhibitors of the third independent of hexokinase glucose signaling pathway.
Yeah. Thank you.
Another thing, you consider that the conjugated ABA form, the glycosyl ester of ABA, of abscisic acid, is uncatabolite. I don't agree with you. I think that the conjugated form of ABA is the main transport and storage form of ABA.
And
the storage, the storage place, the storage subcellular compartment is the vacuole. And to my opinion, in fact, in this regard, there are important missing gene candidates. For example, the ABA GA glycosyltransferases responsible for glycosylation, or also the beta-glucosidases responsible for the release of the free active form of ABA. If you have to begin your PhD thesis research, are there other proteins and their respective genes to complete the list of candidates involved of maintaining of ABA balance in cells?
ABA in cells. If I'm sorry, can you repeat a little the last part?
If you have to begin now, PhD research, are there other proteins and their genes to complete the list of candidates studied concerning the balance of ABA in the cell?
The ABA cell, okay.
还有没有别的基因可以影响 ABA 的合成？
Yeah, we have another experiment. Not in my thesis. It's using the 2016 sample in 2 leaves, 6 leaves, and 12 leaves. We do RNA-seq at 59 days, and from the result, we didn't find many genes different expression, but we want to test VBG1. I think it's the genes may related to the I'm not sure I remember correctly. ABAGE to ABA. I'm not sure. But we really analyze the genes reported to from ABAGE to ABA, and some genes from ABA to PA or DPA. But we didn't find a bigger difference between the treatment. So in our study, I didn't test them again.
Thank you. And do you know how the conjugated form of ABA is transported through the tonoplast in the vacuole?
How ABA transported from
the conjugated form. The ABAGE.
Yeah, the ABAGE. I'm not sure about this. Yes.
This is just a suggestion. They are the ABC ATP binding cassette transporters, and namely the ATP ABCD subfamily of these transporters, which participate in the transport of the conjugated ABA form to the vacuole.
Okay, so we also can test the ABCD the transport to test the
It is very interesting because this conjugated form remains at a very high level in your experiments, but the scale to present this between the ABA free form and the ABA conjugated form, the scales are different in your presentation. That means that some effect may be omitted, may be occulted in the discussion of your results. It is always very interesting, and I agree with Sylvie Dinan, that we have to enlarge the number of sugar transporters studied as implicated in the ripening of grape berry. And I think also that the tonoplast localized transporters, TMT, but also the ESL, the early response to dehydration 6-wax transporters, may be used, and there we have also 2 sweets implicated as facilitators to the release of glucose. That means, I think, that we have to explore in a future work, of course, a large panel of transporters, and maybe include the tonoplast transporters. It is interesting to know also that the ERDR 6-like transporter has been enhanced. The expression of the gene encoding this transporter is highly enhanced by ABA exogenous treatment. And this data have been already reported in the literature.
Okay. Thank you.
Now, I have a last question. May you tell us your your project for the future? What do you do in the future?
I'm a little I'm interested in the how the ABA at Réunion to target the berry ripening. I mean, if it's come from the increase of ABA come from the leaves or other part of plant to arrive to berry to target the ripening, it's maybe should singular from berries to other to leaves, maybe to induce ABA synthesis. So I'm a little interested in the long distance singular transport. So maybe I want to find a lab to work on this if I can find. Yeah, thank you.
Thank you. I think that the storage form of ABA is really very interesting to study, but maybe this balance of ABA in the cells through ripening of grapeberry may be related to the release of the active form from the storage one, from the conjugated one.
Okay.
And the pathway from the vacuole to the cytoplasm is really a very short way and not a long distance way.
Okay.
Thank you, Lina.
Thank you.
Professor Dai, as thesis co-supervisor, you better know the candidates. Would you please share with us your opinion and comments?
Thank you. It's a great pleasure to join this presentation. And first of all, I would like to congratulate Lina, who made a very nice presentation today. Together with Eric, we saw the progress, and I believe this is the best part of the rehearsal that we listened. So this is a really great work. You have involved a lot during the last weeks. And of course, all those progress were also based on the valuable comments from the reporters. And I think you already got very interesting discussion with all the members of the jury. So since we already discussed a lot about the scientific aspects, so I may spend a little bit of time to recall a very brief story about the thesis of Lina. So the thesis of Lina started in 2013 with the financial support from the Chinese government. And she moved first to France, quite strange or quite different foreigner country, I think, but with the very nice atmosphere in the lab and with several friends and colleagues, Chinese colleagues. I think she had settled down very quickly. And then we started the experiment immediately at her arrival. I remember clearly that probably after 2 or 3 days of her arrival, she started to work in the vineyard. In fact, this work is registered in a large project where we also have another thesis who already graduated several years ago, where we have some vineyard results. And finally, we determined to separate the work. So we left all the work in the vineyard to Eloise, and we keep all the work in the greenhouse to Lina. And in fact, we made a very similar experimental design as did Lina. So we sprayed some AB in the vineyard, and even we made some wines. And I remember that Eloise and Lina organized the wine tasting for those wines. So it's a very nice experience for this project. And then I think we really experienced several surprise. Some of them are very good surprise, and some of them I will see really tough surprise. So I think the first happy surprise we got, Lina got, is that she got her baby Tong Tong arrived just about 1.5 years after starting her thesis. So this is, of course, a big achievement for her family, but meanwhile, it's very challenging for her to handle all the things around the process to have her baby, especially in a foreign country with difficulties of language, a different system. But I think after all this work, Lina finished both her role as a new mother and her role as a PhD student. I really appreciate all her efforts did during this process. And then the second surprise, I would not say difficult, but it will not that pleasant, is my quit. So I quit about 1 year ago. I returned back to China and I quit lab. So this is a kind of difficult for Lina, of course, because we started the experiment. We have quite a lot of discuss, and then we have to find another director for her. And I would highly appreciate Eric, who accepted to take over this really challenging task to continue the direction, the supervision for Lina. And we have so many meetings during the experiment and during the thesis correction. So I think this is a really great team to finish the work of Lina. And of course, the last surprise, which is a tough surprise about this pandemic, and which arrived suddenly first in China. And when I got a lot of greetings from my French colleagues, and then France, which we tried our best to be helpful during this difficult time. But I think during this particularly difficult period, of course, we can experience many challenges, emotional difficulties, worries about the situation in life. So, but after all those surprises, Lina did excellent work. I think the first chapter has been, first, she prepared her thesis in publications, and her we just got the feedback for her first paper in Australia Journal of Grape and Wine Research, which is rather positive. We are very confident that the paper will be accepted quite soon. And I think for the last two papers, we will try to submit in the following months. And I believe it will be published somewhere, so that we could really share the results with the world about the thesis of Lina. So I think that's almost all the stories that I would like to share with all the members, and express my thanks to Lina, who worked hard who always have smile on the face, who is very generous and patient with other colleagues. Involved a lot of tasks in the experiment. Really a person pleasure to work with. So by the end, probably I will ask you one question, because this is the moment to give a summary about your 4-year stay in France. So my question is, what will be the most important things that you earned or obtained during your 4-year stay in France? Of course, in addition to your PhD study.
During the 4 years PhD study here, I think the most important things I learned from here is a good team community. I think it's most important things for me to learn, because we are in our project. They have a lot of people to help me, and they offer their personal technology and knowledges, and they are really help me. So I really like here. Yeah.
Okay, thank you. That's all for me. Please open your microphone. President.
Sorry. Yes. Finally, Professor Gomès, as thesis supervisor of Mr. Wang, I am giving you the floor.
Thank you, Mrs. President. First of all, I would like to, Lina, maybe you can close just the sound bar because it's quite powerful. Okay. So, first of all, I would like to thank all the jury members to be here today and to have accepted to evaluate Lina's work. And I'm really happy about the nice scientific discussion that we had all together. So now, Lina, it is really a great pleasure for me to be here today, listening and watching you presenting your work. As you can imagine, I won't ask you questions. We ask a lot of questions about your work already, so I wouldn't ask today. Although I got closely involved I got closely involved in your supervising your PhD lately, as Jiao said, because I took over when he left to take his position at CAS, as professor position at CAS. I've come to know your work in details, and I really appreciate it, like it has been said by all jury members. And I shall say that here that I'm very proud of what you did, of how you evolved during the time in the lab and in the fruit quality group here. I really saw you start from a master student and really evolved to a scientist during your period in the lab, and I'm really happy about that. I'm very proud of what you did. I really like to congratulate you for several reasons. The first reason is that I think you are a very courageous person. And as a proof of that, I take that, as Jiao said, you could have done your PhD in China, in a country you know, in a research system you already know. That would have been, I guess, comfortable. But you choose not to do that. You choose to come to a distant country, France, which has a different culture, which language at the beginning you didn't understand. And I think it's really courageous to do that. Integrate different scientific environment, research system. And I really think that you blend in very well. You really found yourself, I think, comfortable in the lab. And that's really, I would say, a source of satisfaction for us and of pleasure for us. I think also that you were courageous because you had to cope again in a foreign country with the lockdown due to the pandemic, the COVID crisis, and that at a critical period of your PhD. I mean, every period can be critical in a PhD, but for you it was when you were rushing to finish your last confirmatory experiments in the lab, and you couldn't come to the lab at the beginning. So it was quite tough, and you were alone with your family in your flat. And you were also started to think about writing your PhD. But in fact, I think you dealt with it quite nicely, really. The second reason is I think that you are a hard-working person, a nice scientist. You are a hard-working person in the greenhouse, the lab, or at your desk. So you really can go from the field to the computer, I would say, in your research activity. So, and I think it's a quality for a scientist. There's been, of course, moments during your PhD where you had a lot of things to do and it was a bit overwhelming. Maybe at the end when you were rushing to write your manuscript. But you really put very hard work, very hard effort to finally deliver your manuscript. And in that, I think you showed another quality that is prized by scientists. It's resolution. You show a lot of resolution and a lot of energy to really achieve your goal to have your PhD. And I think that's really a nice thing. I would also like to take this opportunity to deeply thank Jiao, which is in fact your true PhD supervisor. I arrived at the end when the battle was 2/3 fought already. And I really like to thank him because without him, this work couldn't have been possible. Also, I shall add that you are listening carefully to our remark and the advice the people gave you, which is, I believe, another useful quality for a scientist. Work as a team and listen to the people you are working with. And you had that quality. The third reason I really want to congratulate you, and this is not the least important one, is that you are a very nice person, really. You're always willing to help your colleagues. Your fellow PhD master students in the lab. You are easy to work with and to go on with in the lab and in life also. And if anyone wants a proof of that, they should just go rush into the PhD office and ask the people what they think about you. And I know the answer will be, Lina is really a nice person. Also, as Jean already did, I wanted to congratulate you to achieve at the same time to build up your family life with your baby and doing your work. I'm turning because Lina is just there in the room. And really, I want to congratulate you for that because it's not easy. So to conclude, you will now start the great journey into the post-PhD realm, like any PhD student do. And you will enter, I hope quickly, the postdoctoral part of your scientific career. I am confident you'll do it fine. You don't have published paper yet, but as Jiao said, we had positive feedback on the first one, and I can assure you that we will do what it takes with Jiao to ensure that you get the other ones published afterwards. Finally, if I may give you just an advice for the future. Have more self-confidence, because it's really maybe the only thing that you have to improve, I guess. So you should have more faith in yourself, really. And again, it was really a pleasure to have you working here in the Berry Quality Group. And I wish you good luck and all the best for your future career, which I am confident will be a nice one. So thank you very much, Lina, for all you have done in the group.
Thank you, Eric. Now, the public part of our discussion is over, and our jury has to deliberate. I would demand the candidate and the guest to withdraw from the video conference.
Lina?
Yeah.
Can you go out of the room?
Yeah,
please?
yeah.
So I can you use my webcam to show that she's leaving the room.
Par contre, ça enregistre toujours sur YouTube.
Yes, I will stop it.
Yes.
Just I will stop the YouTube diffusion. Just a second. Up.